Hello, hello.
Yeah, good morning, everyone. Good morning, dear students, the backbenchers. Please come forward. आगे आ जाओ भाई. Please get up, get up, get up, get up. Fill up the seats in front. My dear girls, please. Please take a seat in the second, third row. Come on, you guys are the future of country. Don't feel shy.
Good morning, everyone, and very warm welcome to all of you. Today, we are here to behold 48th Sir Edward Mellanby Memorial Oration and 72nd Annual Day celebration of CDRI. So, back in 1951, when our institute was inaugurated, the founding mandate was to get technological independence for the pharma sector in India. So, since inception, CDRI has been reviving her vision, mission, so that she can make herself applicable to the current as well as the future challenges. So, after 72 years, I can proudly say CDRI stands as a unique organization having competence of concept to commercialization under one roof. So today we are here to celebrate this historic day, and it's a great honor to share this hall with such distinguished guests. So to formally begin the function, first, let me see, Dr. Habil Khorakiwala, the chief guest of today's event, is here with us, and the guest of honor, Padma Bhushan, Professor K. Srinivas Reddy, who is the former, sorry, sir. Professor K. Srinath Reddy, who is here with us, and I request our director, Dr. Radha Rangarajan, to kindly escort these two dignitaries on the stage. Sorry about it. That's okay. I request our ma'am to welcome them with the flower bouquet. Thank you, ma'am. Now I invite our director to kindly deliver welcome address.
Distinguished guests, colleagues, friends, members of the press, and my dear students. I welcome you to this very special day in our institute to celebrate our 72nd annual day and conduct the Sir Edward Mellanby oration. This is also the penultimate day of our 1 week, 1 lab campaign, a CSIR-wide endeavor to create awareness of the research being conducted in the CSIR laboratories through outreach programs. I want to extend a very special welcome to our chief guest, Dr. Habil Khorakiwala, founder and chairman of Wockhardt, a global pharmaceutical and biotechnology company, and to Professor K. Srinath Reddy, honorary distinguished professor, founder, and past president of the Public Health Foundation of India, who will deliver the Sir Edward Mellanby Memorial Oration today. Thank you both for accepting our invitation. We are fortunate to have our beloved former directors, Dr. Nityanand and Dr. V.P. Kamboj, present here. Welcome to both of you. I also welcome director of our sister laboratory, CSIR-CMAP, Dr. Prabodh Trivedi, Dr. Alok Dhawan, director CBMR, guests from various scientific and medical institutions in Lucknow, the entire CDRI community, and the press. As you all know, CDRI is a constituent laboratory of the Council of Scientific and Industrial Research and was formally dedicated to the nation on this day, February 17, 1951, by the then Prime Minister of India, Pandit Jawaharlal Nehru. Since inception, CDRI's mission has been to develop drugs, diagnostics, and process technologies for India to make healthcare more affordable. Sir Edward Mellanby was the founder director of CDRI. Although he had a very short stint, he laid the foundations for the Institute's scientific agenda and left a lasting impression. Is there a bit of a feedback? As a tribute to the contributions of Sir Mallenby, the Institute established an oration series in his Can you increase the volume? So the Institute established an oration series in his name in 1976, and this oration is delivered every year by an eminent scientist. Today's oration, 48th in the series, is being delivered by Professor K. Srinath Reddy. He is an illustrious, well-known public health specialist, is widely acclaimed, and what he's going to talk about will surely resonate with the research that we are engaged in, considering our strong focus on affordable, accessible healthcare for all. We're all eager to listen to your lecture, sir. So without further ado, I request Dr. Shruti to take over from here and conduct the rest of the proceedings. Welcome to all of you. Thank you.
So as our director has already mentioned about Sir Edward Mellenby, for the newcomers, the new students who have joined recently CTRI, just would let you know that as a tradition, the annual day celebration begins with Sir Edward Mellenby oration, and he was a British physician and pharmacologist who established the role of vitamin D and its role in deficiency in rickets. His work showed that this fat-soluble molecule can be absorbed from diet as well as from sunlight. He also showed that vitamin A influences the development of bones. And during his scientific career, both as a medical doctor as well as a researcher, he has served as Fellow of Royal Society, and later he was elected as Secretary of Medical Research Council. And as Dr. Rangarajan mentioned, that he served as the first director of our institute and laid the scientific foundation of CDRI. And in this lecture series, we invite eminent scientists who have tried to bridge fundamental and translational research. And today we are honored to be joined by Professor Reddy, who is the founding president of Public Health Foundation of India and currently is serving as the honorary distinguished professor. He formerly headed Department of Cardiology at AIIMS New Delhi. Being trained in cardiology and epidemiology, Dr. Reddy has been involved in major international and national studies on risk factors of myocardial infarction, epidemiology of coronary heart disease, community control of rheumatic heart diseases. I would like to mention that Dr. Reddy is a versatile researcher. He's an inspiring teacher, an experienced policy maker, an activist, as well as he is a person of fantastic sense of humor. So as an activist, he has worked on tobacco control program, and he has advocated appropriate nutrition and health being for all. And under his leadership at PHFI, he has been instrumental in establishing 5 Indian Institutes of Public Health to advance multidisciplinary public health education, implementation of technologies to support as well as strengthen the health system. He was appointed as the first Bernhard Lowen visiting professor of cardiovascular health at Harvard School of Medicine, and he holds advisory position in various national and international bodies, including the WHO. He is the first Indian to be elected at the National Academy of Medicine, USA, and also has served as the president of the World Heart Federation. He is the recipient of Padma Bhushan and has served as an advisor for governments of Orissa and Andhra Pradesh. He is also involved in a lot of philanthropic activities, and he has been associated with the Population Foundation of India, which works in empowering women and men in the area of reproductive health and fertility. He has published a book, Make Health in India Reaching a Billion Plus, wherein he lucidly discusses current issues difficulties, and asserts the need for a state-specific strategy to tackle healthcare needs in different states of India. And today he will share his thoughts on path of science, purpose, and process for the societal good. And with this, I invite you to share your thoughts.
This is fine. Right. Dr. Karakiwala, Director CDRI, distinguished scientists of CDRI, luminaries in the audience, ladies and gentlemen. It's indeed a great pleasure to be here in this institution of great eminence, which is advancing scientific research in the area of drug development for human benefit. I'm grateful in my various capacities as a citizen of India, as a public health activist, and inevitably, at my age, a patient as well. So, as the beneficiary in so many different respects, I believe this is an institution of great importance for India, and it's a privilege to be here in these precincts, and particularly in such distinguished company. And of course, Sir Edward Mellanby, having won his reputation for his work on vitamin D and rickets, is certainly somebody whose life and contributions need to be celebrated. But as I discovered during the course of my reading about Dr. Mellanby, Sir Mellanby for this lecture, there is much more to Mellanby than vitamin D and rickets, and that I shall mention during the course of this talk. But firstly, I think we are living in times which are absolutely exciting in terms of the tremendous pace at which science is advancing in terms of its accomplishments. If the motto of the Olympic Games is faster, higher, and stronger, then certainly science is attaining Olympic heights in the speed in which it is advancing and the kind of transformational entities it is yielding, both in terms of knowledge and products, and certainly strengthening our capacity to advance human development in so many different ways. But what is science? I believe it is a structured, systematic, rigorous, and objective method of inquiry into natural or anthropogenic phenomena. But frequently, science and technology are confused. They are complementary, they are essentially related to each other, but science yields an idea, technology yields a product. And while science prepares the way for innovation, it is technology that ultimately translates it into a usable and socially beneficial product. As Patricia Farah says, and this is a brilliant book which I recommend for everybody to read, Science of 4,000-Year History. Science is not just a final product, such as a theorem, chemical, or instrument, but is an integral component of society, interwoven with industry, business, welfare, government, and medicine. Therefore, while scientific research may sometimes take you down the path of esoteric exploration of ideas and knowledge, and that is perfectly justifiable, we must also look at ultimately where that exploration is going to lead us to in terms of social benefit. And therefore, the translation of that knowledge into products becomes an important element as well. And Patricia Farah says, taking advantage of scientific discoveries entails making political decisions about how to use them. And that is where how we have to look at society is ultimately going to benefit from the products of science, or sometimes has to be shielded from some of the ill effects of science which has gone astray. Science, I believe, has a social mission. Science is sterile if it lacks a social purpose, and it must connect with public policy, and public policy in turn will crumble on clay feet if it does not stand on the strong base of sound science. And it is this interrelationship that must be respected and reinforced as we move along our developmental trajectory. But then, when we look at science, ultimately, in terms of research, our objective is to try and come to the closest approximation of truth. We can never find out what is the absolute truth. Absolute truth is meant only for the divinity and like-minded clinicians who believe they know everything. But nevertheless, it is only the closest approximation of truth that we can come to. So we start with an observation and try and move to an inference, but during the journey we are buffeted by a number of forces that interfere with our identification and appreciation of what might be the truth. We have to deal with chance, which is the eternal alternate hypothesis. We have to deal with bias, which is systematic error. We have to deal with variance, which is inherently property of most things, and confounding, which is an additional way of looking at bias, but it relates both the exposure and the outcome in a way. And while we are dealing with all of this, now we are seeing an additional factor that is also beginning to affect the integrity of the observation and the truth, which is fraud in medical research. And this is something that we have to keep alive too, and looking at how we can detect fraud with our own radar and then try and eliminate that influence as well. But then we look at internal validity, which is how the study itself has yielded information on how an exposure is related to the outcome and how valid is that conclusion after we have stripped it off all the obfuscating influences that I've mentioned of chance, variance, confounding, and bias. But then we also have to look at external validity, which is generalizability of the results. If your study is so narrow in its domain of focus of the population included, then the generalizability to other population groups might become limited, and that is a matter of concern that we have to address as well. Then, of course, science lives and dies also by the canon of replicability, because one study itself may not necessarily spell the truth because it could have arisen by chance alone. So replicability becomes an important element as well. But I believe one of the most important statements about science has been made by that philosopher of science, Karl Popper, which is falsifiability, which is that the method of science is to assemble available facts to make a bold conjecture on the basis of those and then gather evidence in a systematic fashion to disprove that hypothesis, not to prove the hypothesis. So the scientist dons the mantle of objectivity and says, look, I think this might be the case, but I'll try my best to disprove it, and I'll only accept it as being possibly the truth if I cannot disprove it. And that's why you have the null hypothesis. And it is that objectivity that we must retain all through as we're pursuing our scientific mission. Now, however, from hypothesis testing, we have come to effect size estimation. Firstly, in terms of, we ask the question of statistical significance, which is assessing the play of chance. Could this observation be the truth, or has it arisen by chance alone? What is the probability by which this has arisen by chance? And that's your p-value. And then you correct it for alpha error, where you might have actually discerned what is a positive result because of multiple reasons, one of which, of course, is that you might have had too many experiments or adventures with too many variables, and many list of variables on which you have done significant testing means increases the play of chance, and you might, by chance alone, get what seems to be a positive result. That is your alpha error. In fact, it has been said, very rightly so, that the more p-values you see in a statistical table of a published trial, then the greater the health hazard. So essentially, that's a challenge that we have. The other element, of course, is that if you inflate the sample size tremendously, then you can get a very minimally clinically significant value also seeming to be statistically highly significant, and that you may overblow the importance of that. The beta error, of course, is where we do not have sufficient power to examine the hypothesis, and we may incorrectly reject the hypothesis, incorrectly reject the possibility that there could be a value. In fact, incorrectly accept the null hypothesis of no difference. And that is essentially a matter of ensuring that you have adequate sample size. A former president of the American Statistical Society said there is also a type 3 error where you use precisely the right methodology to answer the wrong question. So you have to definitely define your research question very well as well. Now, in terms of clinical or public health significance, we've moved to a different gear. We start looking at how are we assessing the impact, not just assessing whether it's statistically significant or not. And the question that we ask ourselves is, even if true, how much would it matter? And therefore, we look at the effect size, and then we look at the confidence interval or the uncertainty band around it, and then say, okay, is it worth pursuing or not pursuing? Is it worth translating it into product and practice or not? And again, when we are looking at where we want to intervene to try and reduce the risk of a particular disease or an exposure which might cause clinical or public health concern, we must also understand the relationship of risk and how the causal inferences have to be made. We are very familiar with the assertion attributed originally to Louis Fisher that association is not causation. But we have then derived ways in which we try and progress from our observation of association to a causal inference. In infectious disease, of course, it's the Cox postulates, but Cox postulates do not apply well to non-communicable diseases, and therefore the Hill's criteria came in for non-communicable diseases in multiple ways. That is biological plausibility, the question of dose-response relationship, and of multiple other criteria that are there in which Hill enunciated the criteria for identifying causality in cardiovascular disease and other non-communicable diseases. Now, in terms of a continuum of risk, we also understand that risk does not necessarily occur across arbitrary thresholds. We are far too comfortable in saying hypertension, normotension, diabetes, no diabetes, obesity, no obesity, and define it across arbitrary thresholds. But now we realize that risk is a continuum. As you move across the continuum of blood pressure, the risk continues to rise. As you move across the continuum of blood sugar, the risk continues to rise. As you move across the continuum of body mass index or waist-hip ratio, the risk continues to rise. It is for you to decide at what thresholds you may want to clinically intervene, but the risk is definitely operating across a continuum and not across arbitrary thresholds. But there can be tipping points, as in the case of climate risks, or there can be interactive risks like cardiovascular disease. If I'm only having high blood pressure, my risk is somewhat different. But if I'm a smoker, if I'm a diabetic, if I'm also having high body mass index, and my age is above 60 and I'm a male, my risk is going to get compounded. So how do you calculate the absolute risk or comprehensive risk? That matters. Then, in climate science, we have also seen just not linear relationships, but cyclic relationships of risk, of escalating risk, particularly in terms of climate change and global warming. Relational risk, where the two layers of the atmosphere can influence each other. Depending upon how much carbon dioxide is there, the other layer of the atmosphere also gets altered. So that's a relational risk. So we are understanding risks in multiple ways. And what we have learned is that it is dangerous to predict risk only using one model. And that we have learned in climate change, in weather, and in COVID. How many of the models that you have seen during COVID prediction have proved to be accurate? On the other hand, you probably, if you will be coming closer to the truth, if you combine the various models, which look at different data sets in different ways, and then have an ensemble model of risk prediction, that actually reduces your inaccuracy. And therefore, when C.P. Snow wrote that attempts to divide anything into 2 digits should be regarded with suspicion, I wholeheartedly agree. But there is also a way in which you have to assess the benefit and the risk of any intervention. So we have been comfortable with relative risk, but that is inaccurate because relative risk is only looking at the risk intervention of the exposure group and the control group, but doesn't necessarily say what is the total quantum of risk reduction in the individual concerned. Because again, you could easily, by reducing the incidence of an event from 2% to 1%, you can say relative risk is 50%, but clinically it may be meaningless. But on the other hand, you want to define what the absolute risk is, and then see how much the absolute risk is altered. Then at the population level, you want to know what is the distribution of that risk factor in the population and how much that can be influenced. That's the population attributable risk. Unless we start understanding the distinction between these types of risk, we will apply them inaccurately. Then in clinical trials, of course, we come to number needed to treat. How many people will have to be treated to reduce an adverse event? Then what is the incremental benefit over the pre-existing interventions? What is the incremental cost effectiveness? All of these are issues that we have to grapple with as we pursue our translational research. Even in diagnostic tests, we recognize that there are very many elements. Very often we have rested satisfied only with looking at sensitivity and specificity, which only talks about how a test performs in a group of people. Doesn't tell you how the test is applicable to the individual patient sitting in front of you. For that, we try and use positive predictive value and negative predictive value. But the problem is, positive predictive value and negative predictive value are very vulnerable to prevalence of the disease in the population. And therefore, that can also not necessarily give you the right information. We also look at the receiver operating curve and look at the area under the curve, which balances sensitivity and specificity, which is a good way of looking at diagnostic tests, particularly in a continuous variable. But in order to find out how actually a test result really relates to the person sitting in front of you as a patient seeking your opinion and advice, you have to look at likelihood ratios, a positive likelihood ratio and a negative likelihood ratio, and then look at the pre-test and post-test probabilities of the person having disease or not having disease, and that's the right way to look at it. But we also can look at, as clinicians, decision failure rate. Okay, what happens if I don't do this test? How many cases will I be missing? How many wrong management decisions will I be making? If that is minuscule, then there's no point in applying the test. Basically, the canon of diagnostic testing is, if your management decision is not going to be influenced by the test result, one way or the other, there's no point in doing the test. But these are all things that need to be understood in the context of research methodology. And this brings us to the Bayesian approach. Nate Silver was a remarkable person who was initially doing the baseball games prediction. And he was so good at it that he started predicting electoral results in US. And he predicted the presidential elections so accurately, virtually in almost all the states, and considered to be a genius in that sense. And he says very rightly, information becomes knowledge only when placed in context. Without it, we have no way of differentiating the signal from the noise, and our search for truth might be swamped by false positives. And that's why we come back to something that was there in the 18th century. 1761 was forgotten till about 30, 40 years ago, the Bayes theorem of conditional probabilities, and looking at how the prior probability and the test result together influence the post-test probability. So that becomes a very important element. Louis Fisher, who brought in the frequentist approach to statistics, ensured in the first half of the 20th century that Bayes' theorem was suppressed. But it has made a big comeback in the last 20 or 30 years, and that is where we must look at even clinical trials of drugs in that perspective. Because, as the t-shirt slogan says, statistics means never having to say that you're certain. We have uncertainty which is reflected in two forms. What's called can't know, which is the aleatory uncertainty, which is because of the randomness of things. Because of that randomness, you cannot really know how a thing is going to behave. But there is a don't know uncertainty, which is epistemic, because of gaps in knowledge about the phenomena and the process of the thing concerned. And that can be filled by research. So you have a prior probability of the parameter, then you gather data, which gives you the likelihood ratio, and then you go to the posterior probability as a product of the prior probability and the likelihood ratio, and then resolve your epistemic uncertainty. And why is this important? Because it is coming up in clinical trials in a big way. Artificial intelligence now uses Bayesian networks. Julia Pearl from UCLA says, and this is as far back as 1988, probabilistic reasoning in intelligent systems, in that book, she says, the Bayes' theorem can be used as a conditional probability to go back and forth between cause and effect. And Josh Tenenbaum from Massachusetts Institute of Technology says, if you can describe your knowledge that way for all different things you're interested in, then the mathematics of Bayesian inference tells you how to observe the effects and work backwards to the probability of different causes. In clinical trials, we are seeing increasing application of Bayesian theory in adaptive trial design, in phase 1 dose-finding study, for particularly the continuous reassessment method, or CRM, for determining maximum tolerated dose of a drug. We are seeing it in phase 2 and proof of concept studies. We are seeing in seamless transition between phase 2 and phase 3 trials in the same trial process. We're seeing it increasingly applied to post-marketing surveillance and to meta-analysis. And we are seeing it in the platform trials, the adaptive trials that are coming up now, particularly during COVID. They've become the norm, whether it's recovery or solidarity. A platform trial is a type of prospective, disease-focused, adaptive, randomized clinical trial that compares multiple simultaneous and possibly differently timed interventions against a single constant control group. So you're no longer doing independent studies with different control groups, different trials. You're actually making the trial process much more efficient. As a disease-focused trial design compared to an intervention-focused, platform trials attempt to answer the question, which therapy will best treat this disease? We couldn't have had the kind of drug discoveries and drug validations in COVID in the time that we had without adopting this methodology. So Bayesian statistics in medical device trials, if you have prior information, then you can get away with a smaller sized or shorter duration pivotal trial. Without prior information, you have to go to adaptive trials. It helps also with have analysis of complex models, adjustment for missing data, sensitivity analysis, multiple comparisons, and optional decision making. This is the Bayesian decision theory. And that is why it's important that we shift our gear from frequentist statistics to Bayesian statistics when we're looking at clinical trials. Now coming to, but I also asked myself the question while preparing for this lecture, mentally. Now we said that there is so much of effort that goes into science and research, but so often things appear to happen by accident. Penicillin being the quoted example of how the fungus came floating in through an open window into Alexander Fleming's laboratory when he was away on a summer holiday. But that may not necessarily be the truth. First of all, the fungus, now it's said, actually came from the laboratory of a colleague one floor down through open corridor. It didn't come through the window. But anyway, there's the romance to the entry through the window. But the Nobel Prize in 1945 went to Fleming, Flory, and Chain, and then Dorothy Hodgkin in 1964 for the crystallographic elucidation of structure. And then you had Alexander Fleming, a bacteriologist, used the penicillin mold versus Staphylococcus aureus. Then the question, of course, has now come up, was he really the discoverer? Because there was a Frenchman, a young physician called Ernst Duchesne, who in 1896, as a part of his doctoral thesis, published in French a beautiful thesis in which he showed that from Penicillium glaucum, he could actually inject into the mice and ensured that they did not die either from typhoid or from Escherichia coli injections. And this was a published doctoral thesis. Because he was just 23 years old, it did not make waves. Even the Pasteur Institute did not accept his thesis, acknowledge his thesis. And he, of course, he went into the army and did other things. But then this came up much later, and in 1949, he was honored posthumously. And he said, this is molds versus bacteria. How the molds can actually, they compete for life on Earth, and they fight each other, and I know it's a beautiful thesis. But if he had published in English, story might have been different. Penicillin might have entered the First World War rather than the Second World War. Who knows? Anyway, in terms of but then came Howard Florey, a pathologist. But one of the important contributions of Howard Florey in Oxford was to assemble a multidisciplinary team. He brought in the whole idea of team science. Till that time, it was all individual scientists going around looking at single author papers and looking for individual glory. But he brought in the whole idea of team science and scaled up production. Ernst Chain, he hired Ernst Chain, a biochemist, and then he brought to Florey's attention the papers published by Alexander Fleming. And then he chemically characterized penicillin. And of course, Dorothy Hodgkin identified the structure of penicillin by crystallography. But here comes Mellanby. That Edward Mellanby, as Secretary of the Medical Research Council, was responsible for this and more this than one. When Florey was being selected for this position in Oxford, there were two external referees appointed, Mellanby and Sir Robert Muir. And Mellanby's train was late. Robert Muir, who didn't care very much for Florey, wanted safe British hands to handle it because Florey was an Australian. And the other candidate was selected. But then Mellanby's train came, he rushes into the selection committee meeting, makes a pitch for Florey, persuades the selection committee to appoint Florey. And then, later on, when Florey wants now to do research on penicillin, having discovered Alexander Fleming's paper, the first grant of £1,600 was given by Mellanby as Secretary of the Medical Research Council. And that Florey leverages to get £5,000 more from Rockefeller Foundation, and there starts the history of penicillin production. So there was a limited yield of penicillin, and Florey goes to US Army to get US Army and scientific help of the Department of Agriculture. And then after searching for different sources of good penicillin yield and identifying, including some molds from India and China as well, they find that the best actually is right now in their neighborhood from a chance finding of a laboratory technician called Mary Hunt, who goes and finds a rotten cantaloupe in the nearby market of Peoria and then brings it to the lab. And then in that, you find penicillin has the best yield, and there starts the huge penicillin production for the Second World War. So then, of course, you have other methods of industrial production. So my lessons from this are very simple. That firstly, it is important not to ignore young researchers. Just because Dushesne was 23, it should not have been ignored. Second is, it helps to publish in English. Third, team science is very important. And fourth, international collaboration is also very important when necessary. In science, the credit goes to the man who convinces the world, not to the man for whom the first idea occurs, said Francis Darwin. And therefore, we need to progress from idea to implementation. Implementation research has recently gained importance. The question is, how do the benefits of scientific discovery and innovation reach the intended beneficiaries? And this is absolutely important because otherwise we find a large number of products piling up without utilization. That's called the secondary translational barrier, and we must avoid that technology pile up. There comes public health. Science discovers, technology develops, public health delivers. You need a strong and robust public health system in order to ensure that vaccines and drugs are properly delivered, whether in community settings or hospitals or individuals or populations. But what COVID-19 has taught us is about complexity, whether it is the transmission dynamics, droplets versus aerosol, virus frequently changing form and variants, vaccines being customized, drugs being developed, non-pharmacological interventions like masks, whether the policies are being enforced or practices being practiced by the community, international cooperation or vaccine nationalism, behaviors of individuals and groups, and unknown factors. Complexity does not permit dependence on linear models. And therefore, we recognize that complex systems are highly composite, built from a large number of interacting subunits, and further subunits are often composite in themselves, and their repeated interactions result in rich collective behavior that feeds back into the behavior of the individual parts. So the key premises of complexity thinking are a large number of dynamically interacting elements, nonlinear interactions, wide diversity of elements, emergence of new elements, new variables from these interactions, unexpected variability, path dependence, self-organization, and resilience. Now, complexity and chaos are partly related but not identical. Complexity requires simple interactions between a large number of subunits and ultimately gives a dynamical behavior. Chaos is the generation of complicated, aperiodic, seemingly random behavior from iteration of a simple rule. It has much fewer interacting units than complexity. So amidst this complexity, we must advance knowledge for societal benefit. Knowledge must be translated into action with demonstrable impact, but the impact also must be equitably distributed across all sections of society. So we realize that the spectrum of science is reductionist in content, but holistic in context. Just like the individual colors of the rainbow may appear very attractive, but they have to be fused together to produce the brilliant white light in which you can see the truth. So the purpose of health research is to provide evidence-informed, context-relevant, resource-optimizing, culturally adaptive, and equity-promoting recommendations for policy and practice. And this, if it has to be translated into policy, requires multidisciplinary research. We often think that as scientists, whatever we have discovered must be taken up avidly and immediately by the policymakers. But even enlightened policy requires scientific credibility from biomedical, epidemiological, and clinical research, but it also requires financial feasibility. It requires health economics research. Operational stability. Can this be introduced into the health system and successfully delivered? It requires health systems research or implementation research. It requires political viability. Is the community ready and receptive for this intervention? Are the clinicians ready to accept this particular recommendation? Are the policymakers themselves ready? That requires social science research. Therefore, you require multiple streams of knowledge to come in, and it's not easy to bring that knowledge together. As Edna St. Vincent Millay wrote in a poem, upon this gifted age in its dark hour, rains from the sky a meteoric shower of facts. They lie unquestioned, uncombined. Wisdom enough to leach us of our ill is daily spun, but there exists no loom to weave it into a fabric. But we can now use a number of ways in which we can combine big data from multiple streams of knowledge and advance our knowledge into translation. But even for guidelines which are developed on the basis of debate between experts, then disseminated to various constituencies who deliberate upon its application and accept it and apply, and then provide the feedback, and even as new knowledge comes in, the guidelines are revised. However, the problem with guidelines is end users are engaged very late in the process, whether they are practicing clinicians or communities. And unless you bring them early into the process of guideline development, you won't get the buy-in, you won't get the successful application. So for this, you need partnerships. Partnerships create a synchrony of effort and a synergy of effect by aligning interests and pooling resources. The challenges to healthcare are access, costs, and quality, how best to deliver value to patients. We need to design and deliver universal health coverage. There's no doubt about it. And this is a target of the Sustainable Development Goals. And we measure the impact of the financial protection provided by universal health coverage by asking, has it reduced out-of-pocket expenditure? Has it reduced catastrophic health expenditure? Has it reduced healthcare-related impoverishment? And for this, the WHO comes up with a cube, because we know that you cannot deliver all of this overnight. So they say there is progressive universalization, in which you actually have 3 dimensions: population coverage, how much of the actual population is covered by the program of universal health coverage, what is the service coverage, what are the services included in the benefit package, and what is the financial protection that is provided in terms of reducing out-of-pocket expenditure and reducing poverty induced by healthcare expenditure. Of course, there are dynamic tensions here between different perspectives. The politicians would prefer to see wider population coverage because greater the number of grateful beneficiaries or voters. The clinicians would like to see as many things included in the service component because they say my patient deserves the best, whether it is cardiac transplantation or cochlear implant. My patient must get the best, or expensive chemotherapy for cancer. Whereas the health economists and the policymakers will say, how do we actually ensure poverty reduction, reduction of out-of-pocket and catastrophic health expenditure, but with fiscal prudence so that we don't bust the budget. And for this, we need to have a benefit package which is periodically revised based on the disease burden as well as cost effectiveness, but also we must recognize that equity becomes a very important element. Equity operates in 2 dimensions: horizontal, where everybody gets part of the benefit package, gets the benefit package, like primary and secondary care. But there's also a vertical dimension where vulnerable groups who are previously disadvantaged and have high health equity gaps are specifically provided additional resources or additional services. And we need to balance all of these. And here comes the role of medicines. It is very clear that medicines are a major driver of out-of-pocket spending and financial hardship, and there is considerable evidence from different parts of the world, from Latin America and the Caribbean, Europe, Southeast Asia, and financial hardship and public health spending is considerably driven by the expenditure on medicines, and unaffordability is also a key driver of foregone care. The people don't even access care, leave aside afford it because of the financial barrier. And you have the great example of soposbuvir for hepatitis C treatment, and look at the cost differentials across different countries. It's amazing to see the cost differentials across the different countries. Then, of course, you have now replete with headlines, whether it is insulin being short, or cystic fibrosis not being able to get treated, or cancer chemotherapy being unaffordable. we see these screaming headlines now coming up from every part of the world, including UK, USA, everywhere. And therefore, when we are talking about insulin, incidentally, insulin, when it was discovered by the Toronto group, they sold their patent for $1 for public good. And now, in the United States of America, people are foregoing insulin or rationing insulin because they're unable to afford insulin. Now, the same problem that comes in terms of anti-cancer drugs, in terms of biologicals, in terms of so many other ways in which access becomes a major challenge. And we have generics, but we also have trade agreements which are prohibitory. Public health protects against trade barriers. The TRIPS agreement has been modified by the Doha Declaration, which gives an opportunity for us in public health interest in order to waive patents or relax patents, and that becomes an important instrument where public health has to join force with the pharma industry and with the policymakers. India's journey to become the pharmacy of the world, product patenting was introduced in 1971. And just as a personal aside, may I say that my father, who happened to be at that time the Minister for Company Affairs, co-wrote that legislation along with Mr. Mohan Kumar Mangalam, who was the Minister for Industrial Development. It helped that both of them were lawyers and both of them were good friends. But that transformed the Indian generic industry. And we have seen how Indian generic industry has advanced. And of course, we moved to product patenting in 2005, but we have now built a very robust industry which serves the world. And therefore, when I chaired the high-level expert group for the Planning Commission in 2011 for universal health coverage, one of the key recommendations we made was that we must provide essential drugs free of cost through public financing and pooled public procurement of quality-assured generics. Of course, we must invest in innovation, new drug development as well, but I think India's strength lies in its ability to provide drugs which are affordable and quality assured. To conclude, the adventure of science is a very interesting one. Carlo Rovelli, in his 7 brief lessons on physics in 2014, writes, quote, "Our hunter-gatherer ancestors in Africa were tracking the antelope to try and hunt it down, scrutinizing and deducting from the details of reality in order to pursue something that we can't see directly, but can follow the traces of the footsteps. In the awareness that we can always be wrong, and therefore ready at any moment to change the direction if a new track appears. That is our ability to correct ourselves, and the strength of science is self-correction. But also knowing that if we are good enough, we'll get it right and find what we are seeking. That is the nature of science. And since you are celebrating science in its best of its investigative spirit and in the quest for its applicability for social benefit, thank you very much. I thank you for your gracious invitation and for your generous appreciation. Thank you.
It was a delight indeed, sir. You reminded us that science without social relevance is sterile. And for a scientific community like us, it is important to prioritize the societal needs while we design any kind of rewarding innovations. So thank you so much for this insight. And we have a few minutes to spare, and any comments, remarks, or questions from the audience, sir is happy to answer.
In that context, I wish to ask you.
Fathers since are being visited on the children. That's dangerous. Okay.
No, but an aspect that is crashingly important at this time in public health is the role of the insurance service industry, which you have not addressed in the disease. Would you please caution us to be very, very careful of making that mistake of thinking that we have we are providing health coverage when we are actually providing insurance coverage?
I'm entirely in agreement. If you read the 2011 report that we submitted to the Planning Commission, we debunked the idea that health can be delivered through insurance. Essentially, we wanted public financing. And even if the insurance industry comes in, it has to be to align itself with the overall structure of public financed and public governed universal health coverage. Leaving it entirely to private insurance is abandoning the people. And that's not right. And that we are seeing in the United States, we are seeing elsewhere too. But therefore, that I'm very clear about, and I fully agree with you. And that is a caution that we have struck a long time.
Thank you, sir. May I now please request our chief guest, Dr. Habil Khurakiwala, to kindly felicitate our orator, and I request our director, ma'am, to accompany. Thank you all. This was our Sir Edward Millenby Memorial Oration. Now we take a quick tea break for about 10 minutes, and we reassemble for further celebration of annual day. Thank you. Okay, I presume all of you have settled down. Welcome back. And now we proceed to the next part of our annual day celebration. I invite our director to present consolidated annual day report of our institute. Ma'am, please.
Distinguished guests, colleagues, friends, students, ladies and gentlemen, it is my distinct honor and pleasure to present the annual day report of the Institute, coming after Dr. Srinath Reddy's brilliant presentation. I'm afraid the annual report summary is going to sound dry and dreary. My apologies for it. My objective is to give you a flavor for what we've accomplished in the last year and some future directions of where we want to go, and acknowledge the support and valuable contributions of colleagues and other stakeholders. I want to start by conveying my gratitude to Dr. D. Srinivas Reddy, Director Director IICT, and at having additional charge of IIM Jammu. He was interim director of CDRI from February 1, 2022 to 5th September 2022. He maintained the pace of ongoing programs, initiated several new activities, and he provided me valuable guidance during my initial days settling down into the institute. My thanks also to all the predecessors of this institute who have contributed to making this institution the way it is today. We are privileged that some of those stalwarts are with us in the audience, and I hope for their continued support. So this is my first annual day report to all of you, having taken charge in September. I present the accomplishments and success stories of the Institute with utmost humility. They are the culmination of efforts of many individuals and many teams, and would not have been possible without the support of CSIR and other scientific agencies, as well as our industry partners. At CDRI, we have a vibrant culture of research, which includes basic science aimed at understanding disease pathology, as well as translational work aimed at therapeutic or diagnostic solutions. We work in 8 areas, which includes infectious diseases and non-communicable diseases, in line with the mixed disease burden observed in India. We also have an interest in developing cost-effective and sustainable process technologies for API manufacturing. And in addition to these research and development activities, we have a large PhD program with more than 450 students currently enrolled. Thus, our institution must balance multiple priorities without losing focus of its mission. In terms of our current pipeline, the Institute has a healthy pipeline of drugs for different disease indications. We currently have 6 clinical stage compounds. The first is umifenavir, a drug repurposed for COVID-19 and licensed to Medizest Private Limited, which is running a phase 3 trial at the moment, and Picroliv, a phytopharmaceutical that is being developed for non-alcoholic fatty liver disease and is entering a phase 3 trial with support from ICMR. We then have 4 drugs which are ready for phase 1 studies. Sentinhale, a novel dry powder inhalation product that consists of rifamycin and isoniazid and is entering an academic clinical trial for TB. This is being developed in collaboration with King George Medical University with support from ICMR. S007867, my apologies, for thrombosis, and S007-1500 for fracture healing are NCEs, and they have received IND approvals. We're working with our industry partners to take these forward. L-ormeloxifene for female contraception is the L form of centchromin, our best known drug. CDRI and CIPLA are co-developing this compound. We hope to move this into clinical studies as well. The pipeline also has 5 NCEs at late stages of preclinical development or lead optimization. This includes S0111793 for malaria, 96261 for leishmaniasis, CDRI 4-105 for chemotherapy-induced neuropathic pain, S0161348 for colon cancer, and S017622 for hyperlipidemia. These compounds are funded by CSIR's IND mission program, a highly focused initiative to accelerate the development of the most promising compounds from the CSIR institutes. In the area of phytopharmaceuticals, the institute has several advanced leads, including Nimetli 118AF1 for stroke, Nimetli 118WFA for bone health, 4655K09 for hypertriglyceridemia, and gibberellic acid enriched fraction for benign prostrate hyperplasia. I want to express my sincere thanks to the project teams and the project leaders for their diligent efforts on all of these programs. I'm particularly grateful to the team leaders for adopting and following new process guidelines that we have introduced for the streamlined progression of compounds. Yesterday, we launched a new product, Green R, a DNA staining dye, in a ceremony with our industry partner, Biotech Desk Private Limited. The founder of the company is here with us, Dr. Shraddha Goenka. Green R is a nucleic acid staining dye with wide applications in life sciences research, medical diagnostics, and biomedical applications. This is an example of a successful public-private partnership. Not only does it provide a more cost-effective alternative to Indian researchers, it is also contributing to the local economy. And I'm pleased to say that this product is now available on the government e-marketplace GEM. Congratulations to Atul Goyal, Naseem Siddiqui, and the team of BDPL for a highly successful endeavor. CDRI is currently part of 5 CSIR mission projects, including the Pan-CSIR Cancer Program, the Antiviral Mission, IND Mission, Phytopharmaceutical Mission, and Immunity Mission. These mission programs have helped us build specific capabilities and human resources in focused disease areas. They have well-defined deliverables and are helping us strengthen our preclinical pipeline. The institute has also received significant budgetary support in the form of grant and aid from different funding agencies. We now have a total of 30 grant and aid projects with total approved cost of 16.38 crores, which have been initiated. I want to especially highlight the CSIR scheme, which is for promoting opportunities for women in exploratory research, aptly given the acronym POWER. And I take this opportunity to congratulate our women scientists, Dr. Divya Singh, Dr. Smriti Vaduria, and Dr. Vinita Tripathi for successfully obtaining these grants. I will now talk about our key accomplishments in terms of knowledge creation and intellectual property. I'm happy to report, as per current data, a total of 220 research papers have been published by our researchers in 2022 with an average impact factor of 4.7. Congratulations. The list of journal publications includes Nature Communications, Matrix Biology, Molecular Cancer, ACS Catalysis, etc. I congratulate all of the authors for their outstanding work leading to these publications. During the year, the Institute filed 6 patents in India and has been granted 2 international patents and 4 Indian patents. This takes our total patent portfolio to 1,170 applications. Congratulations to all the inventors. In terms of our PhD program, 42 students submitted their thesis and graduated in 2022. I wish them a fulfilling career ahead. We also welcomed 94 PhD scholars on campus. I wish them a successful innings at CDRI. Welcome. Under the Skill Development Program, 242 participants from 102 colleges covering 21 states of India had received training at CSIR-CDRI. Of these, 175 were women and 67 were men. And we continue to have a deep interest in our skill development programs. Our institute has added certain new facilities during the last year. I'm happy to report that the sustained efforts of Dr. Amit Mishra and his team. With his efforts, the institute received approval for a drug testing laboratory. This will primarily serve MSME customers and startups needing to evaluate the purity of APIs and finished products for their regulatory filings. In addition, the complete infrastructure for manufacturing oral solid dosage forms and inhalable formulations for IND submissions and clinical trials is now available through CRTDH, a dedicated facility within CDRI. This further extends our end-to-end capabilities in drug discovery research. Congratulations. In a recently held surveillance inspection of CSIR-CDRI's GLP test facility by a team from NGCMA, the facility successfully completed its recertification with an expansion of scope to include in vitro mutagenicity studies. I thank Dr. Sharad Sharma and Dr. S.K. Rath for leading this effort successfully. Another institutional initiative I want to tell you about is the Gender Advancement for Transforming Institutions, the GATI program. This is a pilot program initiated by the Department of Science and Technology to promote gender equity through the removal of structural, social, and cultural barriers which prevent stakeholders from achieving professional excellence. Some of the activities we are undertaking under the GATI program are creating housing for married women students with children, upgradation of the creche facility, workshops on gender sensitization, mental health support, and a placement cell to help students with employment opportunities. I thank Dr. Neeti Kumar for leading the GATI efforts, as well as Dr. Ritu Trivedi and Dr. Sanjeev Yadav for playing a key role in student-related activities. I'm happy to report that during this past year, CDRI received the STEM Technology Transfer Impact Award 2022 for the technology titled Standardized Nanoformulation from Spinacia oleracea for Osteoarthritis on 16th November 2022 from the Society for Technology Management. I congratulate the entire team of innovators for this unique achievement. Friends, there were many individual rewards that our scientists received, but in the interest of time, I'm not going to go through each one of them. I congratulate all of the awardees and wish you many more accolades in the future. I'll now give you a brief on the financial status. For the current financial year 2022, we have generated a reasonable amount of external budgetary resources, similar to previous years. During the year 2022, a total of 26.29 crores were generated, which includes 22.46 crores through external cash flow and 3.82 crores through laboratory reserve. Special congratulations to all my colleagues whose efforts led to the generation of these funds for the Institute. Thank you. The Institute has received financial support from the Department of Biotechnology, Department of Science and Technology, Science and Engineering Research Board, SERB, the Indian Council of Medical Research, and the Ministry of Health. We also received valuable support from the Director General of Health Services and Family Welfare and Medical Education, Government of UP, and we have received support from KGMU, SGPGI, and KEM hospitals, particularly for conducting clinical studies. I want to acknowledge and thank all of these supporters. There are several industry partners who have engaged in fruitful collaborations with us, such as Biotech Desk Private Limited, Medizest Goa, Cipla Pharmaceuticals, Mark Laboratories, King's Herbal, Zydus, and many, Themis Medicare, and several others. Our thanks to all of our industry partners. I'll now briefly comment on some future directions. We have made significant progress in the last year, and I celebrate our wins. While our basic research continues at a good pace, it is clear that we must step up some of our translational activities. Let me offer you a global perspective. Today, India supplies 20% of the world's drugs, but in terms of value, we rank 14th. And coming in the close heels of the lecture that we heard in the morning, while I agree that we cannot count our success merely in terms of value, and that impact is measured in many other ways, I still think that we must look at this number with some with a view to understanding what is actually happening. What it means is that our manufacturing sector for drugs is performing very well. The truly innovative products that generate value are still elusive. And the reason to chase some of these innovative products is not only for impact in terms of diseases that pharmaceutical companies may not want to work on, but also because one must build in sustainability. So how can we make, and it's not to say that innovation is not happening. It means that innovation perhaps is happening in pockets and in bursts. So how can we make success consistent and repeatable? And how can we do this at CDRI? I envision 3 prongs to the CDRI strategy. One is to focus on a limited number of translational products for very well-defined unmet clinical needs while ensuring clear differentiation from products in the market. Whether this is to generate value or this is for sheer impact, it does not matter, but we must focus and we must have a very clear purpose. The second, we must build strategic collaborations with industry and academic partners so that we can advance in synergy together. Today we get stuck. Today we find that we can go from point A to point B, but then the road ahead all the way to point E is not clear. So we must assemble our partners at the very beginning with understanding of achieving milestones and moving on. So this is part two. And the third part is optimized decision making based on pre-agreed criteria for the progression of compounds, so that decision making is not biased, decision making is streamlined and efficient. Simultaneously, we must work on improving our administrative functions. This includes reforms in procurement, upgradation of facilities, streamlined data management, revitalization of our technical and administrative cadres, and strengthening the infrastructure for our students. These will require cohesive efforts by all of us and a belief that we can make change happen, and we must dare to make change. We have enormous support from CSIR headquarters through our DG in particular, Dr. Kalai Selvi, who has been very supportive, and I'm sure that we will have support from her in implementing a reform agenda. I'll now end this report by acknowledging some of our current and former colleagues. In the year 2022, 24 of our colleagues have superannuated after a long association with the Institute. I wish them a happy and healthy life ahead. I report with sorrow the untimely demise of one of our colleagues during the year. I pray that her soul rest in peace. I express my sincere gratitude to all the advisory committees of CDRI for their valuable support in addressing the myriad institutional issues that come up from time to time, for which they contribute time, effort, and mindshare so that we may put in place effective policies and implement them. Really, thank you so much. I'm grateful to all my colleagues for their cooperation at many levels. I'd like to specifically acknowledge Dr. Nevedya Chattopadhyay, Dr. Atul Kumar, and Dr. Ravi Shankar for their support with a diverse range of administrative matters. I also want to thank senior colleagues, Dr. Sharad Sharma, Dr. Saman Habib, Dr. Atul Kumar, Dr. S.K. Rath, Dr. Amit Mishra, Kural Srivastava, Dr. Sanjay Batra, Dr. Atul Goel, Dr. Gautam Panda, and Dr. Shashidhara for their wise counsel, support, and contributions to institutional activities. I thank all the area coordinators and heads of division for their support in managing our research programs, the heads of project management, business development, administration, finance and accounts, stores and purchase, and their teams for steering the activities of the Institute. My special thanks to all our students for their contributions to the research and development work of the Institute, and you're all here, thank you. Finally, a big round of applause to Dr. Sabyasachi Sanyal and a very, very dedicated team of scientists and administrative staff for organizing today's activities. Thank you all for a patient listening, and I look forward to a great year ahead. Thank you.
Thank you, ma'am. It's always a pleasure to recite the accomplishments. It not only keeps the spirit high, but also helps to keep the spark alive to do more. So thank you. With that note, now I invite Dr. Bhavana George to introduce the chief guest of today's event, Dr. Habil Khorakiwala, to the assemblage. Over to Bhavana.
Dr. Habil Khorakiwala is the founder and chairman of Indian multinational pharmaceutical and biotechnology company, Wockhardt Limited. Wockhardt is a global pharmaceutical and biotechnology organization providing affordable, high quality medicines, next generation antibiotics, vaccines, etc. It is India's leading research based global healthcare enterprise in the fields of pharmaceuticals, biotechnology, and a chain of advanced super specialty hospitals. When I was reading about Dr. Khorakiwala, something that caught my attention was his mantra, vision to execution. His leadership enabled the organization to become global enterprise. Dr. Khorakiwala also pioneered biotechnology research in India and introduced indigenous human recombinant insulin and later its analogs. His efforts have significantly impacted availability of affordable medicines to the masses. He is an alumni of Purdue University and Harvard Business School. Dr. Khorakiwala has been Chancellor of Jamia Hamdard University and past President of the Federation of Indian Chambers of Commerce and Industry. He has been recipient of several renowned awards. Purdue University awarded him an honorary Doctor of Pharmacy degree, Shiromani Vikas Award for his outstanding and inspiring contribution towards national development by the hands of Mother Teresa, to name a few. He also constituted Wockhardt Foundation, an organization with a structured and completely dedicated approach to societal needs. His achievements cannot be summarized in such a short time, so I will stop here and invite Dr. Habil Khorakiwala to please come up on stage.
Very good morning to all of you. Your director, Dr. Radha Rangarajan, Professor Srinath Reddy, who gave an outstanding memorial lecture. And I have learned few things from what he said. And he established a little backdrop of giving an overview so that one can focus on a very specificity which I intend to do. I'm so happy to see Dr. Nityanand here after many years. And he is really the pioneer and legend for whatever one sees CDRI, CSIR today. Your director has provided you pathway and priorities, which I was listening. And she has a very interesting background, that she has an academic background and worked with Rockefeller and Harvard, and then started her own organization of research, and now joining you. So that probably she gives new perspective, new eyes to look at it, and that is what she has set her priorities. And I wish her and all of you a great future of taking CDRI in a new pathway. Let me first, what I intend to do today is two things. One, very briefly give you a pharma biotech perspective of what has happened and where we are in a Indian sense and a global sense. And then take one of the molecule we are doing research, because this is yours is a research organization. So I thought I'll share details of our antibiotic research which we are undertaking. So I think if we look at our industry, we are fortunate that, like very few industries, it is generally led by all the companies which you see across, excepting few, by the first generation. And therefore, the industry has the kind of vibrancy, I would say, entrepreneurship, risk-taking abilities, and that is what one saw over the years. And I think a major change came from the Patent Act, which you mentioned earlier. Process to product patent, that is where the whole Indian industry flourished. When I joined, 80% of business was with global pharmaceutical companies, multinationals. Today, 90% of business is with Indian companies. So that's a transformation which has taken place. Second major change took place when the liberalization started in early 90s, and the new Patent Act was likely to be enacted in 95. And some of the industry leaders thought, what do we do? Because now you are getting a global challenge, both in terms of business model and in terms of research also. So from a business perspective, a lot of us started, because of liberalization, you can do business outside India. They went all over the world. And that is how we established today, 2/3 of our, 60% of our is an export led. And there are large companies, many companies have bought companies, established the businesses there. And the research, I will talk to you, us and few others have initiated research, drug discovery research and other technology research in 90s, late 90s. And all these led to a relatively high growth for the industry over the last 30 years or so. Now, what I would like to focus on VOCAT only, which I think VOCAT, very nice word spoken about us, of what we have done and achieved, and thank you for that. But if briefly speaking, what is our mission? The tagline of the company is "A life wins," and that is, sometimes you have a very simple tagline, and what it does is really creates focus, inspire people, and we have to make a choice. They know what is important. So if life is important in our pharmaceutical, healthcare, hospitals we have, so people take a decision in favor of life rather than other conditions when those challenges are there. And that has a very long sustainable purpose and brings whole organization together into a approach or a theme where there is, they can talk to each other on this basis basically. And I think our focus has been basically the science, commitment has been our focus of whatever we have done over the years. And this is something, just let me tell you a little bit about the antibiotic program. Okay. We entered this area of antibiotic about 25 years ago. And one of the thought, why antibiotic, why not diabetic, why not cancer, why not? So I personally looked at in a competitive sense, because when you do research, it has to be competitive. And unless it is competitive, you don't get anything out of that research. And my of our colleagues also did it, but they took a path of where the more research is there, where the more value is there, like diabetes, cancer, other areas, all of them. We chose antibiotic mainly because there were 2 or 3 considerations. One, we saw that big pharma have vacating this space because it was not as remunerative for them for whatever reasons. And I saw a 20-year trend where the drug approval in Western countries were coming down year, every 3-4 years data you look at over last 15-20 years when we took this call. Second, I think India had about, even today also 10-12% of pharmaceutical usage is antibiotics. And our thinking was, at least we go into this area, we will have the medicine available for India and other emerging market. If we have it for global markets, nice, good, but at least there is a idea of sustainability. And that is where we started. And the third is that there are so many therapeutic area which were there. And currently a global market is about 43 billion and India is about 22,000 crores, which is roughly for 1/3, 10-15% of total market here. This is very interesting. Sometime about 5-7 years back, this Jim O'Neill, who was the British economist and the Secretary of Exchequer in UK. He was also earlier chairman of Goldman Sachs. And he studied this whole idea of scarcity of antibiotic to meet the resistance strain. And resistance were developing very rapidly. And he came out with a report as an economist saying that if there are no new antibiotic discovered, by 2050 it would be a major cause of death worldwide. And trillions of dollars of economic cost would be there. So that was the scary thing. But the assumption was that there is no new antibiotic coming in. And our view is obviously that to prove him wrong. And with the new antibiotic, we will not have this kind of scary scenario which he created. That's it. Now, this is very interesting to know. These are recent data. And this came out of the CDC UK. I mean US. They have studied the incident, we know all incidents of COVID over the last 2 years. And in 2 years, we lost 6.7 million lives worldwide. And these antibiotic resistance, which is there, half a dozen organisms which have the resistance like Pseudomonas, Acinetobacter, Klebsiella, Oxaliate, Metallobacter, and others. We are losing about 4.9 million lives every year. Now, the COVID context, all of us know every day we were in news, people dying. Many of our friends and relatives lost their lives also. But simultaneously, we have lost more lives because of antibiotic resistance. 4.9 million per year means twice that number, nearly 10 million. Okay, I haven't got this right every time. Anyway, see, antibiotic, if you see, is one which cures a problem. If you have an infection, you take antibiotic, it cures. Many of our diseases, if you look at it, it manages the problem. Diabetes, heart disease, cancer also does not cure. To some extent it cures, it can reoccur also after some time. Somehow it manages. So therefore, from that point of view, that's one important aspect of antibiotic research. I don't know why I'm getting twice the same. Okay. And obviously, as we know, there are two kinds of organism. One is in a community acquired and another is a hospital acquired infection. And these are respiratory and diabetic foot ulcer are generally community based. Pneumonia, bloodstream infection, bone and joints are, and many others are hospital. So therefore, discovering to my mind, a novel antibiotic is a fairly monumental challenge. One is an economic challenge because it is not commercially as lucrative as many other diseases like oncology or cancer or other areas. At the same time, because of the kind of protocol one requires in a hospital, for any other disease, you use a new drug if it is better. Here you keep it reserved till other drugs don't work, older one, then only you use antibiotic, and that's why it's a both challenge. And I think there is something wrong. Gandhiji, I just like to quote him, if we have the belief that we can do it, we shall surely acquire the capacity to do it, even if we may not have at the beginning. And that's why exactly what happened with us. We were starting a drug discovery program for the first time, but we thought this is a good area to get into it, and that is what we started the journey. And what we have done over the years is a build up an organization which is a multidisciplinary, both for drug discovery, clinical, later on, and all aspect of research with various facilities what you require, medicinal chemistry, your animal house, microbiology, pharmacokinetics, whatever you need. We have created a very comprehensive end-to-end research capability over the years. And I must say, right from the beginning till now, all our scientists, some of them are coming from your institute also and working with us, are of Indian scientists. We don't have a single foreign scientist right from the beginning till today working in our discovery lab. For clinical, obviously, we had to take, we have a team in US and Europe, which is helping us along with people working here in India. So it is a fairly, and I must say that we were looking in a competitive sense, who are my competitors worldwide? And we found that Big Pharma has exited, they just buy molecule from individual small companies. So in reality, we do not find a single company who has globally a kind of a comprehensive drug discovery program for antibiotics. We are only talking of bacterial antibiotics, we are not talking of all infectious disease basically. And I'm sure your institute has been working on drug discovery program, and this is a generic approach. Okay, you design it, it takes 12, 15 years before you start identify and complete a whole drug discovery program. Now, one of the important things which our team has done over the years is there are a lot of competencies and capabilities we do not possess. But for research, you require that kind of, like, for example, you need what are the organisms available worldwide, whether your drug is sensitive against all the organisms. Like Western countries like US, and they want, when you make an application, you have to test all the, if you're making a claim, okay, works on say Pseudomonas or Acinetobacter, to look at all the organisms available now within last 3 years, whether it is sensitive, where the resistant. So, you know, those, and therefore we have, our team has collaborated with about 15, 20 institutions. And that is a part of that ecosystem and networking which is necessary for to be really competitive on a global basis. To find a drug, and in fact, we have already licensed out one of our drug to a Chinese company, 4873. Which is oral drug, gram positive, which is for pneumonia and other things. So now if we look at what is a WOCAT, what are the strategic pillars of this drug discovery program? First, obviously, is whatever we want to do, whether it is clinically viable. In a sense that if you work on this area, whether the product finally will be there, it is superior to whatever is available. And that is fundamental to any drug research, because you have to predict after 5 years, 7 years, 8 years you would be there, whether you would be over. So you need to understand what others are doing and what will you be doing to that extent. Second, I think our approach has been how do you de-risk the project. So in order to de-risk the project, I think we didn't try to go fundamentally on very basic research, but we use whatever knowledge available in this space. And sometimes many of the dots of knowledge are not connected. And that is what our scientists tried to do, to connect unconnected dots. And that is where I found our main drug 5222, which I will talk a little later on. We got a new as an enhancer. Inhibitors were there, beta-lactam. So that is where they got a synergy concept that it enhances dramatically, and that is how we saw that it works on some of the most difficult resistance organism, which I mentioned earlier. So that was one aspect. So our main drug, we have a drug with a single molecule like 4873, 771, they are single molecule. But our main drug 5222 is a combination. So it's an older drug, cefepime, and our new drug, zidabactam. That is our drug discovery. And collectively it works better. And surprisingly, when you look at zidabactam per se, it is a very poor antibiotic. But when it is combined with cefepime, it's like bursting in terms of activity. Obviously, mode of action I discussed, and we have also established, I must tell you, a global KOL network, and that is very, very important. Because if I have to fund and sustain, how do I know our team, what they are saying is there? They also have to know what they are doing is competitive and people buy it. So we have created this global network of opinion maker for last 15, 17 years, and they were meeting. So somewhere around 12, 13 years back, I thought, okay, let me come and attend, because we have been there for last 10, 12 years. And do I see some ray of hope at the end of long tunnel? The first time I attended this meeting, this was in Boston, in Chicago it was. And we had about half a dozen top KOL who are involved in this antibiotic research from Harvard and Cleveland and other institutes. They were present. And it was a 3-day conference. So first, when I looked at the people who were there and they are prepared to spend 3 days, itself said something. Okay, whatever work was done, they had some kind of respect for that work actually. And I realized at the end of a 3-day session, okay, they were contributing and participating in terms of the way forward we should do. And this is a program we are continuing it. We had every year a 3-day conference with the KOL. Some are same, some are additional, depending on the program. So that played also a very important role for us to know, okay, what we are doing is worth doing and it is enough differentiating from a science point of view. Now, if you look at the program which we have, if you look at gram positive, gram negative, I think all areas are covered in this. We have got 2 of these gram negative drug. MROC is already launched. The 4873 phase 3 getting over in next few months. We'll be introducing in India by end of the year, early next year, once we receive approval. 5222, which is our main drug, which I'll elaborate a little bit, is already under phase 3 clinical trial in Europe. We have not yet started in India, but we will be starting soon in India. And we expect next 12, 15 months it will be over. And then it is a global trial. So we will have patients from US, Europe, China, India, and few other countries. That is there. And then we have additional 2 or 3 drugs like 6777, where we are in a phase 1 clinical. And actually NIH, because we thought, okay, we will delay phase 1 because there's so much we are doing it in terms of management of resources and everything. So they found NIH is doing phase 1 for us, and they are spending $8 million because they feel there is unmet need. This is an antibiotic which is once a day. It's a combination with zidabactam and ertapenem. And there is another drug which they mentioned here, which is an oral drug which would be a enhancer. And it fills a big vacuum because there is no oral antibiotic which has come in last 30 years, which is really effective. And this raises the whole level of effectiveness to a very different level. These are the molecules which we have at various stages. I did mention in clinical trials, all of them. And these are the pipeline basically, where they are there, each of them, at various stages of phase 1, phase 2, phase 3. So we have 3 drugs in phase 3 actually, out of 4 we are talking. One in 6777 is in phase 1. And that 4873 is getting this year, 5222 we might complete clinical trial next year. And let me talk to you a little bit of now, just to get you an idea of the US FDA gives QIDP status of a molecule, and that really means that it is unmet need is significant. Second, it has a fast track clinical pathway, they agree when you share the data. Like 5222 is a single arm study. They normally want 2 arms for a new molecule because one of them was, and safety data you have and they've seen. And also approval is faster. They do it in half of the time than normal new product approval. So we have received so far 5 products fast track approval QIDP, and you can see all the other companies have either 1 or 2 of them. So this is a globally competitive space. Now, let me just elaborate. MROC, which we have launched in India, which is a gram positive for a drug. It has no side effect. It works even in the orthopedic conditions where you have the film formation and antibiotics don't reach. So it works there, and it does not have the side effect of the known drugs like linezolid or vancomycin, which are generally used as a staph. Then this is our drug 5222. It's been there for around quite some time. We don't see any drug, excepting Shionogi product is approved, but it has a significant side effect, and it has got a black box warning from the US FDA for approval. So it has to be used restrictively. Our drug is extremely safe. And also it already resistance have developed because it is not as it works basically, but it self develop a resistance because the dosage is not good enough for not making resistance, because you need a correct dose of antibiotic. They couldn't do it because the drug they use has the toxicity, and that's why they got a black box warning. And if they'd increased the dose, it would not have been approvable. And we have good patent, variety of patents are there, and we have patent till mid 30s, 35, 37. Yeah. The very important thing about 5222 is it is in a phase 3 clinical trial, and 3 or 4 patients, 3 patients so far have been treated under compassionate use. We are doing a trial for urinary. We would have to start afterwards for HEP and VAP. Now there was a patient here in Lucknow, Medanta, a very serious Nepalese lady in 40s. She was on ventilator and tried all the antibiotics available here worldwide, which are recently approved, and nothing was working. So finally, the doctor asked for DCGI permission to use for compassionate use. We provided the drug. She was off ventilator on the 3rd day, went home on 10th day. And And as the protocol, they followed up the patient for 1 month, so they don't have afterwards side effect, and that was clear. Then there was one 12-year-old boy in Chennai Global Hospital. Same situation. Permission, FDA, DCI didn't delayed 1 or 2 patient permission. And unfortunately, what happened? The patient died. Because in antibiotic, you can't delay. Instead of clearing 24 hours or maximum 48 hours, they delayed by 5-7 days. And I wrote it to them, okay, look, thank you for giving some permission on time, but we have lost a couple of lives which we could have saved. Then I think they have come to the speed and now they are looking at that, that is corrected, because this was the first time of experience. So the system takes its own time. So we are very happy that these are, both were Pseudomonas cases where existing antibiotics are not working with anything available. Now, this is something important, that unless you get properly protected through patent, there is no way you can invest money into drug discovery, which is a long-term investment, like we are there for 20 years. If you didn't have good patent, global patents available, there is no way we could even invest money. There's no return after doing so much work, investment and time and a commitment. So we have filed all these drugs are well patent protected in a global basis. So briefly, to say that their fundamental strategy is innovation, we are balancing it. As I said, we de-risk it, we try to do what is possible to do and not go to totally unknown area where there's a lot of knowledge sitting. Many times just connect some of the knowledge. This enhancer Zidabactam is result out of my Dr. Mahesh Patel, who is my chief, and he is there with the other important thing I must tell you is my drug research team over last 25 years in terms of leadership position has not changed. That is mainly, I believe, they need an environment, they need an ecosystem of understanding them and supporting them when it is needed. Second is, if they see that they are getting a drug, there is a self-motivation taking place. Because also many of you are scientists, you have been working here for many years and decades. Sometimes you spend the whole life, 30 years, and you don't find a drug in the market. Here, as a scientist, you see something happening that is there. And third is our KOL concept, our idea of interacting with a large number of other institutes wherever we needed help, also gives them a different breadth, rather than what is environment within the organization, but outside the organization. Then that is a kind of interactive process. There is a lot of learning taking place. There is a lot of self-growth one perceives, and they get excited about all these things happening. And that is where the team, we have been able to have that. Obviously, product differential and clinical and commercial viability is the essence. So this is in brief of my talk. And obviously, this is fundamentally what WOCAT stands for: impacting life, saving lives, affordability, and the creating value for all our stakeholders. So thank you very much for giving me this opportunity. I enjoy talking to you, and this is a picture of our research lab in Aurangabad, a drug discovery research lab, where we have other research also, biologicals, pharmaceuticals, and everything else. So, pleasure talking to all of you. I know there are all the scientists working here. Large number of students are there who are studying here. So, I hope that whatever I have shared inspires you to continue what you want to do in life. Thank you.
Thank you, sir. If we have the belief to do it, we surely will get that capacity to do it. Okay. Sure. It's open for discussion.
Sorry? Yeah, I can tell you what I will have a price in US. India would be a fraction of that cost. Today, new antibiotics which have come, which are not comparable, they are roughly at $1,200 per day. So it will be roughly 12,000, 10-day treatment would be $12,000. We will have a fraction of a cost that I told you here. Yes.
Makers are willing to do that, politicians are willing to do that, academicians are willing to do that, and industries are also willing to participate. What do you think, in your opinion, are the main hindrances to proceed further in this direction?
See, I was president of FICCI, and this was the main, at that time it was a very hot topic of public-private partnership in all aspects, not only in research, but all aspects. And there were some training centers which were there, which was a government. They wanted private people to come and do that. It didn't work out at the end of the day because the government bureaucracy, the system, I'm sure you run through that experience so much. So nobody wants to give away anything. When you have a partnership, it's both sides. So that is the major problem. We tried also when Mr. Modi was chief minister. We come from Gujarat in Palanpur. There was a civic hospital. So we chatted. He said, I suggested, okay, if something I can do. He says, there is a civic, why don't you manage? So I said, all right, we'll try. I said, even if I lose money, it doesn't bother me. He said, I'll give you the budget, what we have. You can do, make some paying patient and do whatever. And if you need more money, you put it. I said, okay, I'll do it. I spent 2 years, I put our people. We could make a head or tail out of this. We just couldn't move forward. So fundamentally, I believe that public-private partnership physically of this kind is not workable. But one can create new models of public-private. In industry, private sector will not change, government system will not change. That is a reality. So we can think of a different model of public-private partnership. And in healthcare, what I think government is doing is a different kind of public-private partnership. What they are doing is they are going to provide free of cost to treatment, hospitalization, various, and they fix the price. You go anywhere, they can do at this price, it is available. So government pays for it, and they are using public-private resources. So one has to innovate in terms of creating this public-private partnership of this kind of a model, where you do not try to put existing organization physically into public-private. It has tried 30 years, all the government has tried, it never worked. But this kind of synergy of using private resources, funding public health or education, that is something one can seriously think about. Yes.
Thank you very much, sir, for giving us the inspiring story. And actually, we are working on drug conjugation instead of drug combination. And I would like to know what is the global interest on drug conjugation and drug editing in terms of drug combination.
I don't think I'll be able to comment because research is not technology driven or pure science driven. Research is driven by the vacuum or the need of unmet clinical need. So fundamentally, our research has to be driven not, you can use technology to go and identify unmet need. But you can't say, if I'm doing a recombinant technology, what is the scope for it? Or if I'm doing stem cell research, what is the scope for it? Obviously, there is a scope, but then the issue lies in identifying what is my clinical gap or need it is there.
The other thing is, how is the future of this editing already existing drugs approved?
That's a new science. No, today the new drugs. Today, if you look at it in recombinant technology, stem cell science, editing, and so on and so forth, that is where 70% of new drugs approved are today. 10 years back, the biologics had about 20% of total dollar value. Today it is 50%. And tomorrow it will be 70%. So it's a matter of time. The new science is biology, because that is where the potential for unmet need is much greater. The science is moving towards that. Many of the areas in cancer research is biological today. I must tell you one more thing. We had a meeting with the Prime Minister about three or 4 years back, and he had called half a dozen people from industry to discuss. No secretaries, nothing. He was alone there with one person to take notes. So we spent 2 hours. We were discussing various things, how, because pharma is, you said rightly, pharmacy of the world, and even in US we made a significant 50-60% of prescription in US is our generic products. So where do we go from here? And what some of us told him basically, okay, whatever you help research grant and others you have withdrawn, so it's okay. But one thing which is important is the innovation and new science must be encouraged, basically, and that is where India will remain competitive. We have the capabilities to do that in all fields. So one way or the other, and then I think he took it seriously. He arrived, and the one more thing which still happening but very slow happening is, I said, we follow regulatory approval process. We follow what West does. There is no fundamentally fresh eye, because sometimes the West has a history, the bureaucracy of 20-30 years of regulation, and it's not possible sometimes to change that. So they have one system. I think we must have science-based, smart regulatory, because when you have what to create, like we look at drug discovery or any business, you say, what is my competitive advantage? As a country, what is my competitive advantage for research and innovation? So I should be science-based, but should be a smart regulation, which unnecessary removes a lot of steps which are not scientifically necessary. And still you don't compromise the basic science. Because what is happening, which we are observing, the regulators in the West, the whole regulator is a totally de-risk mechanism. They want so much safety data, so much this, little thing happens. There's a big lobby of big pharma and big biotech companies there. And they create fear concept, if you do this little change, this will happen. So as a result, the regulation becomes more and more complex. We have to use science, but use it in a smart manner and simplify regulation. And that is where we can create competitive advantage. And that is where we get drug discovery, innovation taking place. And India is a very big market of 1 billion plus people here. So even if you introduce in India and approve a drug, there are a lot of people who will take that product. Because for pharmaceutical, West is an important market. But that's not the only market. 6 billion people live outside the Western world. So for treating any medicine, anything, the whole world is a market. If you have, that is how you can create innovation. Thank you very much.
Thank you so much for that wonderful talk, sir. It's always a pleasure to learn from industry perspective. Now I invite our guest of honor, Professor K.S. Reddy, and our director, ma'am, to kindly felicitate our chief guest. So now that all the three of you are on stage, I take this privilege to felicitate some of our CDRI team members who are celebrating the Silver Jubilee year at our organization. First and foremost, I would like to request Shri Jaiprakash he's a senior technical officer and he's from Division of Engineering Services. Sir, I invite you to kindly come and receive the honor. I would like to call upon stage Shri Siddho Hemram, a Senior Technical Officer from Division of Engineering Service, to take the token of appreciation. अब मैं बुलाना चाहूंगी श्री मोहित कुमार शुक्ला फ्रॉम डिवीजन ऑफ इंजीनियरिंग सर्विसेज, ही इज सीनियर टेक्निकल ऑफिसर. प्लीज वेलकम सर. May I now request श्री रामकरण हरिजन जो इंस्ट्रूमेंटेशन प्रभाग से है, आप प्लीज स्टेज पे पधारिए सर. लेट्स वेलकम श्रीमान मोहम्मद इरफान जी, जो जनरल सेक्शन में सहायक अनुभाग अधिकारी है. Hearty congratulations, sir. Now I invite श्री मोहम्मद सलीम जी, जो जनरल सेक्शन में सहायक सेक्रेटरीट है. I just want to say that you have been integral part of our organization's journey and success. We are eternally thankful to all of you. And once again, congratulations for celebrating Silver Jubilee year at CDRI. Congrats. Thank you. Our chief guest will have to leave now. Despite his busy schedule, he came all the way for us to be the part of today's event. Thank you so much for your time, sir.
You let me know. Thank you. Thank you.
It's now time to pay homage to those members of our CDRI family who got superannuated last year. And to do the honor, we have our guest of honor, Professor K.S. Reddy, and our director, ma'am. This year we have 24 members, and I request them to come on stage to receive the honor. First and foremost, I invite Mr. Ved Prakash Mishra. Okay. Next. Mr. Arun Kumar Srivastava, Mr. Vijay Bahadur Lal Srivastava, Mr. Rajender, Dr. Neena Goel, ma'am, Mr. Prem Prakash, Mr. Dilip Kumar Khare, Dr. Gopal Gupta, he was there, Mr. Suresh Kumar Verma, he was a lab technician here and he has served for 40 long years in CDRI. Please give a big round of applause. Srimati Madhu Chaturvedi, Mr. Nandapal Yadav, Mr. Naveen Prakash Mishra, Mr. Bharti Bhushan, he was Senior Technical Officer. Please come, sir. Mr. Sukhdev Prasad, Mr. Pankaj Bahadur, who was our former senior COA, Mr. Mahendra Kumar, thank you so much for coming, sir. Mr. Raju Arora, he was a senior technician for 39 years in CDRI. Thank you for coming, sir. May I now invite Mr. Ramjeet? Very important person. For 39 years of service in CDRI. Congratulations, sir. Please come. Mr. Vinay Tripathi. Yes, sir. Srimati Preeti Rastogi. Srimati Savita Tripathi. Shri Akhilesh Kumar. Dr. Renu Tripathi. Mr. Rajendra Prasad. Okay, that completes my list. All I want to say is words are actually not enough to thank you all for all your service to CDRI. And however, I extend my warmest greetings to all of you. Your service will always be remembered. Thank you so much. My special thanks to Professor Srinath Reddy for doing the honor. Thank you so much, sir. May I now invite our organizing secretary of this 72nd annual day event, Dr. Sabyasachi Sanyal, to kindly propose vote of thanks.
Before thanking everyone, I must apologize for the tardiness in refreshments. I take the full responsibility. But we have now reached the last segment of the program, and it's my privilege to propose the vote of thanks on behalf of the CSIR-CDRI family. We are thankful to Dr. K. Srinath Reddy for delivering the Sir Edward Mellanby oration, and it was a profound talk, sir. Thank you so much. Now, you know, healthcare is a great ecosystem that comprises of patients who often are one disease away from abject poverty, clinicians, academicians, researchers, pharma and allied industry, as well as policymakers. We scientific people often tend to lean towards the more exciting exploration and discovery process. We get a lot of insight from you, sir, for the this wide world that's out there. Thank you so much. We thank Dr. Havil Korakiwala. He's gone now, for his illuminating talk and nice story about 5222. And I'm sure the budding entrepreneurs here, they will take a lot of inspiration and insights from his talk. I thank all the dignitaries, Professor Nityanand, the youngest member of the CDRI family. You always are an inspiration, sir. Dr. V.P. Kamboj and our sister lab directors, Dr. Prabodh Trivedi of CMEP, Dr. Bhaskar Narayan from IITR, Dr. Alak Dhawan, CBMR director. Thank you, sir. Thank you very much. We thank the press and the media for covering the program. Now, organizing this event wouldn't have been possible without the tireless efforts by a lot of members who were coordinating the organizing subcommittees. I'm thankful to all the scientists, students, technical officers, administrative and finance staff for their support in organizing functions. I thank Dr. Sanjeev Yadav and team for the 1 week, 1 lab program. It was wonderful. I'm especially thankful to the canteen people for the arrangements pertaining to the OWOL. Drs. Rajkamal Tripathi, Sarita Tripathi, Mr. Sanjeev Sekhar, Ms. Rashmi Rathore, Mr. K.K. Saxena, Mr. Rishi Kant need a special mention, whose fruits of labor you are going to enjoy in today's evening. I thank the creative team of Drs. Anand Kulkarni and Niti Kumar, that includes Dr. Bhavna George, Shruti Raju, Mr. Rabindranath Londe and Kazim. We thank the tireless efforts by Drs. Kishore Mohanan, Arunab Dasgupta and S.K. Raj. I'm also thankful to our IT team for arranging the online streaming, and the cultural committee and participants who will probably rock the stage tonight. I apologize if I missed mentioning any names. I thank all the student volunteers and audience and each and every one of you. Thank you very much. And let's now rise for the national anthem.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
So thank you. It concludes the program.